5 cents might not seem like much until you're saving 5 cents per cow per day. Then it really adds up. New AminoSure XM precision-release methionine provides the optimal combination of cost, feed stability, rumen stability, and intestinal release to deliver the best cost per unit of available methionine on the market today. Hello everyone, and welcome to the Real Science Lecture Series. My name is Kim Jones, a member of the Belchem marketing team, and today we welcome Dr. Phil Cardoso from the University of Illinois, Urbana-Champaign, to discuss the importance of balancing amino acids in the transition period for health and reproduction. Dr. Cardoso is a professor of animal sciences at the University of Illinois, Urbana-Champaign, where he is conducting research and providing outreach programs in the areas of dairy nutrition and reproduction. His experience with dairy farms in Brazil and the U.S. bring a different perspective and management skills that help him with discussions in class. His broad technical and analytical background is improved every day by the exchange of experiences he has with students, staff, and world-renowned professors at the U of I. He obtained a PhD in ruminant nutrition from the University of Illinois and a master's in DVM degrees from the College of Veterinary Medicine in Porto Alegre, Brazil. Dr. Cardoso was born and raised in Brazil and soon discovered his passion for dairy cows and got involved with livestock operations in the southern part of Brazil. Dr. Cardoso, welcome to the Real Science Lecture Series. The floor is now yours.
Thank you very much, Kim. I hope everybody is hearing me well and can see me well as well. Yep. With lights.
Yep.
Okay, thank you. I would like also to start by thanking Belchem to invite me for this prestigious webinar series. I'm very happy to be here. And we were allotted a certain amount of time that I'll try to share as much information as I can for some of the research we have done here at the University of Illinois, but also using some outside research from our groups to kind of either contrast or validate some of the things and some of the points, and hopefully today, everybody can come up with one thing that they can do different tomorrow, or just making sure that they are on the right path for giving farmers or providing farmers with the best profitability and the best path to success, whatever that means to that farmer, right? It's important, I think, for us to try to identify that as well. Okay, so talking about farmers and trying to identify what is success for them. This is me a few years ago. You don't have to look to the data there, to the year. It's almost 20 years ago. But that was something that I was doing or practicing and actually executing a lot. That was the DAs. So clear a transition cow problem. And we usually would attribute that to, to the negative energy balance going through the transition period. So you can see that when you are relying on pastures, sometimes it doesn't rain enough. Is it too hot? That transition from the winter to the summer sometimes is a challenge for these farmers that would have corn silage on the feed bank, but also would rely a lot on a pasture as well. And that was at that time a fairly new problem, metabolic disorder for them. That's why you see here my friend here, Jairo, he's like looking at me and making sure that that's a real thing. And then I had to show him what's going on. Nowadays, it's something that is very well known. And I think we came a long way on how to nourish those cows, how we make those diets to prevent some of those disorders to happen, where nowadays in the US, it's very rarely that you see a high incidence or prevalence of those disorders like displacement of ovomason. But we focus a lot on ketosis, negative energy balance, and those associations, but also something that you all know from Alan Bell that described then the protein side of things. So, hey, you know what? That negative energy balance, actually, you are seeing the same thing with protein, where cows, they cannot consume as much protein as they needed. And right around that time, someone that you know pretty well, Dr. Tom Overton from Cornell, he published this data where he's showing here true time during the transition period What is an indication? So this is a blood biomarker telling me how much muscle is being degraded in that cow during that period for most likely being used as a source of nutrients. And we always call energy, right? But let's say a source for amino acids. And you can see that around parturition and early lactation, that amount gets increased a lot. So we already knew from a long time ago that cows are mobilizing not just fat or adipose tissue, but also muscle in some regard. And I think our quest here is to design diets where this is minimized, because we believe this is what's going to make the cow produce the most efficient and more profitable milk for the farmer. So here are some of the things that our group here in Illinois, we've been talking about during the transition period that we really need to watch out. But I'll focus today on this topic here that we believe it's extremely important for cows and for the success of farmers. So let's look a little bit in what would be this diet. So you can have here, later on you can pause, you have the presentation, you can look at everything. But like I said, I'll focus on this protein part. So for this close-up cow or one group dry cow, we would talk about 12 to 14% crude protein. But I want to add, and how can we move forward to maybe start looking that number with a little bit less intensity, let's say. And I'm going to do that by showing a research done here in Illinois by Dr. Jimmy Clark and his student at the time, Ignacio. And what they're doing, they collected research papers that were showcasing what is the crude protein in the diet and what is the milk yield that those cows are providing. So you can clearly see that, for example, cows that were eating diets at 18% crude protein, they were producing anywhere from 23 kilos to 45 kilos. And if I just, change the direction of the ellipsis, you'll see that at the same token, cows that were producing 25 kilos of milk, they were eating diets from 12% crude protein to anywhere from 24% crude protein. So it's fairly clear that this is not a precise direction. And that means that nutritionists, they would get very frustrated trying to target a specific milk yield based on crude protein. And by the way, when we get to this, when we calculate what is the most efficient point in this curve, that's where we get to the 18%. So most of us would say, hey, lactating cows, we would be about 18 to 19% crude protein. That's where we should be. That's pretty much based on where that curve starts to flatten out, the most efficient point there. But I know, and I'm pretty sure that most of you know, that it's too much to ask for crude protein and assessment of nitrogen times a constant to deliver exactly how much milk that cow will be producing. So you're very familiar with this graph here, with this figure, where crude protein is explaining this part of the equation, where rumen degradable protein, RUP, that's not described at all. Where microbial protein, and I would argue that we should move to this metabolizable protein. That means what is the amount of protein the cow will be absorbing after digestion. And then I think we should actually go to this part here, amino acids. I'm not sure we are quite there yet, but that's what I'm highlighting here. So we need to understand what is the metabolizable protein getting to the cow. And also, we would have to understand that will require a model for us to understand that. So we're going to have to go through that model, right? Now, one other thing that I want to highlight is that energy, the amount of starch, the amount of fat, amount of fiber that the cow is consuming will drastically impact the amount of microbial protein and therefore metabolizable protein. And one thing that I want to point here and let it in this presentation is that I believe we have very good equations on how to predict in both sides of the world. You go to Europe, you go to US, we kind of agree on how to get to the metabolizable energy. And I think I just want to set that aside. I think we can do a good job in any model how to calculate the energy component of the diet. I think that energy is going to be impacting our microbial protein and therefore metabolizable protein. And I'll just let it sit for a little bit and I'll bring this information back a little further. So how do we calculate MP? So you are seeing MP in your software and you are trying to push to a target. I'm going to tell you a number and how it's, well, here in the US, right, you have different systems all over the world. But in the US, up to 2021, so in 2001, the proposition was, hey, if I know days that that cow is pregnant and also the calf birth weight, I know how much metabolizable protein that pregnant cow will need, dry cow. So fairly simple equation, right? Then most recently, NASEM 2021 comes with a different equation that now it's not really, is really considering this essential amino acid flow getting to the intestine. So remember I told you we need to understand MP, but really what the cow is consuming is amino acids. We should forget talking about protein nutrition. Maybe we should talk about amino acid nutrition because that's what the cow is using. But I guess for a lack of detailing in our diets or feed ingredients, we kind of go back to, hey, just nitrogen, that's fine. But I think it was a good push and we move forward and now understand, hey, what are the amino acids that are coming out of the layer of that intestine? What is the microbial fecal protein that is going there? What is my target for efficiency on each one of those amino acids? And then it's going to get to, hey, all this is going to get to the amount of amino acid that the cows are going to need, and that's going to come back to this MP concept, or all the amino acids the cow you need. However, I want to highlight here, and then there's always a good out of the bad, right? The model doesn't tell you everything, and actually the model tells you that. So for example, for a close-up cow or heifer, this is going to be the amount of crude protein. And what does that mean is that, let's say, depending on the intake of that animal, it will require 900, let's say 1,000 grams of metabolizable protein. However, it's already telling you, if you go read the chapter, it's going to tell you, hey, heifers may need more or less. We don't know yet. And it tells that the model ignores the protein for colostrum, memory development, immune function, and restoration of body protein. They're just saying, hey, we don't have data. I'm not going to say anything about it because then I'm going to be, liable or whatever. I'm not going to put anything. We need more data. So it's up to you. It's up to the nutritionist to use the model as a reference. But that's going to be a number that you're going to have to think about how you apply that. That's why also I think how AI is never going to figure out that job. So that's the good part of it. Artificial intelligence is going to be pretty hard to figure out all the, details from each type of region and everything else. And also the lack of information. So we need to rely on your experiences and other trials that have been performing, like some of the ones I'm going to show you here, that can really focus on not necessarily efficiency, but in performance. There's a little bit of difference there. Efficiency versus performance. Different story there. But remember, we are talking about 1,000, but we are saying, I'm not counting for a lot of things that I believe are really important, right? But they don't have the data. What can I do, right? And then for lactation cow, in the past, if I know how much protein the cow is producing milk, that's it. I would know my metabolizable protein needed. And then very similar, the new model now, it's kind of very similar in addressing, hey, we want to know what is the essential amino acids going there, all the efficiencies and everything else. So a much more improved model on how to give us that metabolizable protein. So I'll give you a very practical experience on approaching formulation diets, right? So I'm using here AMTS, so the CNCPS model. And actually, this is the exact diet that you can find in the paper. All the papers I'm going to talk about here, you can reach out to me, you can reach out to Balchem folks, and we're going to provide you all the papers that you need so you can go in more detail maybe. But this is the screen of the old software that is still available up to some point, not forever. But what's going to happen here is, okay, I can toggle what is the percentage of MP of these amino acids. And that was one of the ways that we started improving the formulation beyond crude protein. This is a dry cow, close-up, pre-fresh diet. Where those numbers come from? Well, those numbers come from NRC that would do, hey, we are adding here this methionine to the cow, and this is what's coming out in milk. Where is the point where there's not, we are just wasting our time or money doing that, or it's not as efficient? And that's where they come up with this 2.4 for methionine, 7.2 for lysine. Using the Cornell model, that was not very different, right? So it's about 2.6 for methionine and then 7.0. So CNCPS, NRC 2001, they were not super, or they were not very off on what they were telling us to feed on those 2 amino acids to start off. You know, the one that was seemed to be lacking the most or more deficient in cows, or they were more required. Then those numbers were kind of very, very similar, right? However, there's one thing. If I'm feeding 1,000 grams of MP, the percentage is going to tell me, okay, I'm there. But however, grams, I may be off, right? So if I'm feeding 1,500 of MP and keep the same percentage as if I'm feeding 1,000 grams of MP, then the amount that the cow is getting is going to be different, and that may be an issue. So then the software, as you click here, you can get the grams. So here is the grams of metabolizable methionine and lysine that I'm feeding to this cow. So here are the numbers. And more recently, then there is this linkage between the grams of metabolizable amino acid, in this case methionine and lysine, and the energy, the mega cow. And again, let's go back to the beginning. The belief is that we can determine energy pretty well. I can determine energy coming to the cow very well. So if I associate that metabolizable amino acid to the energy, I may get a pretty good response. And this is again from the group from Cornell, from Van Amberg, showing, hey, I'm infusing here this methionine, and I'm checking milk, and I'm seeing where is the best efficient to convert that methionine that I'm feeding the cow or infusing the cow versus what's coming out in milk. And that's the numbers that they got. So the numbers 1.10 or around 3, that's what you're seeing here. But if you really kind of do a zoom on that line, you're going to see that some cows, they do the same thing with less. Some cows read the book and they do the same thing with what they're being told. And some cows, they need more to do the same thing. So there is a little of cow variation there, or efficiency on how to use that amino acid. So I will report here one research that they did, 144 animals. So this is all post-peak animals. And they fed those cows for 114 days. And to make it simple here, they have 3 diets. One diet, they call it neutral, and that diet was feeding on the line. So all the amino acids, I'm already reporting here, histidine as well, but they went through a lot of the other amino acids as well, the essentials. And then they formulate a diet to be 1 standard deviation in lacking those amino acids, or 1 standard deviation above that line for the requirements. So imagine that cow that ate more, the cow that ate in the line, and the cow that ate below the line. So you can see here that the amount of methionine or lysine goes up as we go from -1 to 1 standard deviation. So what was the main result here to this? There's a lot of other information there, but cows that went from the negative to the neutral, that was a gain of 3 kilos of energy-corrected milk, versus cows that went from neutral to positive, they had an increase of 1 kilo. Again, this is a research controlled environment. So the idea is that treatments can showcase all the benefits or negative of those treatments, right? But what I see here, and I'll translate to you, is that if you have a farmer that is in that negative, What is that negative? Well, you are feeding about 1 grams of methionine to mega cow, and you bring them to 110, you can expect more results, or the results to show up more often than if they're already at 110 and you want to bring them to 130, right? And I think that's just expectations and probability of things happening in all the different environments with different physical characteristics of TMR and everything. That's what brings. So it's saying that I would encourage everyone to be looking at this and try to be at least here. There's a financial as well investment to go from here to here. So that needs to be taken care of very, very well. But I believe, sometimes the cow's requirement and need for amino acids does not relate to investments, but it's needed. And I would say that neutral is something that we should aim for at the first step, at least. But how about dry cows? So let's talk a little bit more. So crude protein, maybe we don't go there. We recommend 1,200 grams, could be 1,300 grams, but that would be a minimum to guarantee that we have all the amino acids, branching amino acids there for that cow. And maybe still build there a safety factor there, right? And then this would be the recommendations that would give to that cow when associated to energy, right? And this would be for lactation. For dry cows, I'm going to advance here and say we should be looking perhaps at an amount of that amino acid as a gram in a gram basis. And I'm going to tell you why right now, why associated with energy may be a little bit challenging. So one thing is that we want to explore is that you out there, you guys are feeding diets that are high energy, usually no straw in that diet, or you're feeding diets that are a control energy diet, a straw diet, that is feeding what the cow needs, not more, no less. And usually you're going to be adding 3-4 kilos of chopped straw to that TMR. The difference in energy of those diets are considered. And so the amount of amino acid that you would feed would be very different. So this is the experiment. That was my PhD student, Emily, is still here in Illinois. And what we did was, hey, this is the high energy cow, that high energy diet with amino acids. This is a control energy diet that we're feeding to some cows with amino acids as well. And you have a control energy diet without amino acids. And we are targeting all that kind of around that 1,200 grams protein. But the interesting thing on this design is that we are keeping the ratio to energy the same. So to that top recommendation, when we are feeding amino acids, and here is room-protected methionine and lysine, we are keeping the same ratio to energy. That means that I'll feed more methionine in a high energy than in the control energy. Same thing with lysine, right? And if I don't feed amino acids in a control energy diet, that's where usually I'm going to be. So around here in the US, I'm seeing people that they are feeding a diet with inclusion of straw. I would almost guess that they will be about 26, 27 grams of methionine. And I'm telling, hey, I think you should be even a little bit higher than this. I think you should be about 35, 34 on that methionine. And then that lysine should be almost close to 100. And we're going to see the impacts of those treatments. Those 3 treatments, high energy with amino acid, and then control energy with or without amino acids, and everything when we feed amino acids in the same ratio to energy. So one result that I want to show you here, that we were able to follow those cows. So with an ultrasound, we can tell what is the amount of muscle and what is the amount of adipose tissue, the subcutaneous adipose tissue that those cows have in different time points. So we follow the cow from -28 up to 70 days. And I will show you here what we found out here with the back fat only during the prepartum period. So from -28 up to calving. And you can see here a tendency, I would say, that this control energy without amino acid lost more back fat when compared to control energy with amino acids. No difference in control energy or high energy amino acids. And on the same token, this tendency here for muscle depth, for a little bit of gain, but definitely not a loss like the control energy without amino acids. So on tissue mobilization, it seems like feeding the control energy with amino acids is the best way to go for that cow not to mobilize tissue. Now, we divided that from 1 to 4 weeks, and we didn't see any differences. But then from 5 to 10 weeks, you can see that the cows in the control energy without amino acids, they ate less when compared to the cows on the control energy and amino acids. And again, no difference between control energy and high energy on this token here. I'm not going to focus on that control energy versus high energy diets. I mean, we could do another webinar just talking about that. I think you already had other webinars that discuss that topic. So I'll focus on the amino acid part. And here, maybe that's where that high energy starts showing up, why there is like a little bit of issue there. So you see that the energy balance, you can see that this control energy diet with amino acid, they were able to lose less or have a higher or a smaller negative energy balance. And you can see that now this high energy diet is pulling those cows to mobilize more tissue, most likely, when it's coming to this 5 to 10 weeks here. And then the body condition score change, again, something that we saw in that a lot of that research between control energy, high energy, you can see that we didn't see a different statistical difference here, but we saw the big difference here. So out of this body of work that is just coming to publication in the Journal of Dairy Science, we can see that control energy diet without the support of amino acids, it is probably hurting the cow a little bit, right? So if you're feeding a control energy diet and collecting all the benefits on the health side of things, no DAs, no ketosis, cows are just going with the intake, really should look into the quality of the protein or amino acids coming to the cow, and that may need for some additional rumen protected methionine and lysine there. How about reproduction then? We kind of posed ourselves here to talk a little bit about this. So we did work where we fed from prepartum to 72 days in milk. We fed cows with methionine or not. We went through it, and then we superovulated those cows and harvest the embryo. And one outcome of that, we collaborated with Peter Hansen at the University of Florida, where he was able to look into the embryos and say, hey, how much lipid, how much methylation, how much DNA is in those embryos. And then we caught that this blastocyst, hey, the cows on the methionine, they had more lipids than the cows in control. Not that they were fat embryos, but they had more lipids than the control. So another part or subgroup of those embryos, we were able to collaborate with Purdue University, where they have this machine that we put each embryo here in a plate, and the machine shoots a laser, and this laser comes back, and that's where the detector will read what's coming back. So it looks like this, like a heat map, like weather channel type of deal. And according to the color, then you can determine the saturation of the fatty acids that are inside that embryo. And then, of course, it's not a huge range of those fatty acids, but it can tell us unsaturated, monosaturated, and polyunsaturated. So it seems that there is a shift when the cows were consuming methionine for that more polyunsaturated embryo. And okay, remember the embryo, 7 days, is inside the uterus of the cow. At that same point, actually 15, 30, and 72 days in milk for those cows, we were able to harvest uterine biopsies and then do the same work, but now we have a bigger amount of tissue, so we have more ability to determine different fatty acids. And you can see that at 15 days in milk, there is no difference between the cows that were with methionine or not. And you can see as it goes to 30 and 73 days in milk, there is that shift for this more perhaps unsaturated type of fatty acids compared to control. And remember that is we didn't go that far, but one thing that we discussed in the paper is that, hey, remember that this polyunsaturated fatty acids are involved in perhaps inhibiting prostaglandin F2 alpha. Perhaps that leads, this polyunsaturated feeds to cholesterol, and that's going to be important for progesterone and maintaining the pregnancy. That's what we discussed. We didn't measure that, but we kind of hypothesized that's the mechanism on what's going on, and that uterine tissue now, it's talking with that embryo, and there is that exchange of nutrients. Now, another work done by the group in Wisconsin, they fed cows from early lactation until they got pregnant, multiparous and primiparous, and they checked for pregnancy at 28 and 61 days. And you can see that at primiparous, there was no difference in the loss of those embryos. However, big difference happened in multiparous cows. So the multiparous cows, that consume methionine, they were able to keep that pregnancy more than the other ones. So it's kind of feeding from different trials here. And also, one thing that group from Dr. Wiltbank's lab, they were able to actually, in a subgroup of cows, do an ultrasound, measure the size of that embryo, and see that no difference in size in primiparous cows, but a difference in the size in multiparous cows. So there's that association of size and cows being able to signal to the brain of the cow saying, hey, I'm still pregnant here. So maybe that is some of the things that were happening there. And maybe this embryo consuming rumen protected methionine is able to be nourished better and therefore grow and therefore be able to keep that pregnancy. That may be some effects on the progesterone as well that we didn't measure. So how about lysine? I want to touch a little bit about, because I talk a lot about methionine. So we did a trial. This is Laura, the PhD student that was responsible for that trial. And we really want to know, should we feed prepartum? Should we feed postpartum? So that's where this 2x2 factorial here comes, where we have cows receiving lysine the whole time, or only prepartum, only postpartum, or not receiving at all, right? And methionine was at full blown, and we just were changing, adding or not lysine. So here's the diet. So you can see crude protein, not to that high level if you want to check on that. Starch kind of going on that 10 unit points change, trying to maintain that. But here's kind of more to the design. So trying to push to that 1,200 grams. Remember this diet here, no blood meal, could be replicated anywhere. And we are really pushing to that 100 grams of lysine, and control was at 80, and we were keeping the same for methionine. And postpartum, then we went from 143 to 159. Hindsight, I think we should be closer to 180 or even 200. There are a lot of groups that have been exploring those numbers and are getting pretty good success. So I think our lack of response postpartum probably was more related to, not feeding that much. So dry matter intake, there was this tendency. Hey, if the cows ate lysine, rumen protected lysine before calving, there was a tendency that they would eat more after calving, a positive thing that we want. We didn't see any difference in milk yield per se. However, when we checked for energy-corrected milk, fat-corrected milk, all the components was higher, right? So it seems like we kind of discussed a little bit. We didn't have the ability to do mammary gland biopsies before calving, but it seems like the lysine was pretty important in priming that cow to produce in this first 4 weeks after calving. That's why the prepartum impact on after calving. We didn't see, we followed, this is my other student at the time, Annie, and then we follow ovulation, no difference in there. But when we did those uterine biopsies on day 28, we saw some indication of the cows that were receiving lysine maybe had more capacity or ability to deliver mucus into that uterine tissue for that cleaning perspective, and less amount of superoxide dismutase. So the oxidative stress perhaps was into a smaller degree when cows are getting lysine. We also follow the calves of those cows. So we have a whole paper on it. Brittany was the one that did the work there. As soon as the cows calved, we pulled the calves out, gave commercial colostrum, and then we saw that there was this tendency for the calves from cows that received lysine, they had more intake of crude protein and metabolizable energy up to weaning on week 6. So, and there's some other things there, but I'm just trying to bring a perspective that, hey, this pregnant cow is also going to be impacting the calf that is going to be born. We also follow the placenta. This is not a cow in our farm, but it's just an example here. As soon as the placenta was dropped, we then collected parts of this, of the placenta, and then we did analysis where gene expression, where 0 would be the control, and the bars are going to be what's going up or down on the lysine cows. So I'm going to summarize here. There was genes indicating more growth and cell process on the placenta from cows receiving the lysine. There was more ability, or the indication of more ability to gather energy, like glucose, for example, and cholesterol production for the cow placenta from cows receiving that. Less oxidative stress. And interesting enough, as we are adding that lysine, it seems like the placenta is also being able to pick up a little bit more of the methionine that may be circulating on those processes. So it's kind of a balance. It seems like, again, if I add the lysine, now one tissue called placenta is able to capture, or there's an indication to capture more of that methionine. So that was very hard sometimes for us to, go and get a blood sample. Sometimes the blood sample would be lower, but the tissue is utilizing a little bit more, well, I digress. Let's go now to the real world there, to commercial farms. This is a farm here in Illinois, pretty top, solid production, over 7.57. We've done at 4 State now, their conference. We did a little competition. Seems like farmers are being able to get over 8.3 pounds per cow per day, so over 4 kilos pretty much per cow per day. But I think there's a lot of things that they do right. It's not just the feeding for amino acids. I think they chop straw. So if you follow Dr. Drakely and Dr. Overton, I think he's kind of promoting the chopped straw, how important it is. But they do a very good job on their transition period with the dry cows. And yes, they feed blood meal, they feed rumen-protected amino acids, and they take really good care of their forage. So it's not a one thing and, you are there. I think there's a lot of work that goes into having high components. Now, I want to share, finishing up here, I want to share a little bit of experience in our farm. So if I call our milk plant and say, hey, how far back again you can give me our results from our tank? And they told me since 2014. So I have since 2014 here, the bulk tank milk true protein. So if you're talking about crude protein, you should add 0.2 to what the numbers I have here, right? So you can see that through time, our milk protein has been increasing through time. But also you can see that it's not a straight line like this one that I just projected, right? It's ups and downs, and you guess right. Every time you have summer here in this side of the world, in July, you get the reduction in components. And this is happening all over the world, right? We just changed the month that the summer is happening. But that's happening all the time. Another thing is that I can collect or know since when we started feeding for amino acid formulation. That's about 2015. That's when we started formulating based on MP, grams, association with energy. That's the most recent, like I just shared with you. And we have the data from the farms around us. That's our route. So 30 farms that also deliver milk to the same company. So you can see that it seems like we've been able to collect more protein, especially in the winter months. Remember, our facilities are not top notch. So the summer really, hammer us. And then in the winter is when we can perhaps collect a little bit more of those solids. And also a line that tells me the amount of protein from all the farms that are delivering, about 900 farms that are delivering to that milk plant. So one interesting thing is that most of the time, maybe when we change management, then that's a little bit tricky. But most of the time, even in the summer, we don't go as low as our friends. We still are able to keep higher protein in milk than our neighbors or other farms. And then I want to share this last trial here where Russell, my student at the time, we did a trial with heat stress cows and par fed. So these cows here on the blanket, you can see they're being heat stressed, and the other cows are just chilling. The cows that are being heat stressed, the intake is going to go down. So we force the cow that is par fed to drop in the same intake. So again, there is no confounding of intake. If I add heat, the cow in heat stress goes down intake. I drop the other cow the same amount, right? And I'm feeding protected methionine or not to those cows. And we are able here to do a mammary gland biopsy in collaboration with Dr. John Kent at the University of Guelph. We're able to identify all the units, the alveoli that are producing milk, the ones that are actually dying in green, or the ones that are actually thriving and alive, right? And when we put that in a perspective, we can see that, yes, heat stress really impacts that, but there's that interaction with the diet where it seems like the cows consuming the ratio of, one over the other or not. It seems like the cows that were receiving the room protected methionine, then they were able to have less of those cells dying and more of those proliferating when we do the ratio compared to the control. So maybe that's that amino acid nutrition that we push pretty hard in our farm allows our cows not to drop the components as much. And not to talk that at some point we may have this 0.30 difference that for us here in the US, especially in the Midwest, represents money. So if I can get, instead of 3.3, 3.6 protein, that means that my paycheck is going to be bigger. Of course, what is my investment to get there? And that's a whole conversation about feed cost and efficiencies, right? But this other people from Australia would tell me, hey, I don't care about the money. Even if I don't get paid, I should be worried about milk protein. Why? They were looking into farms, 74 farms in Australia, over 359,000 calvings, and they identified they were looking for what is associated with reproductive success in those farms. And what they found was the milk protein concentration. So as milk protein increased, the conception rate at first service, or cows is still pregnant by week 6, was impacted by the amount of milk protein. And the blue and brown here, you can see the difference according to the slope is different on how much milk those cows were producing, right? So there is that association with the embryo that I showed you, that it's needed those amino acids. And here in a different perspective is that, hey, if the average hosting cows is producing 3.2, 3.3 protein, and your herd is at 3, maybe there is a signaling that there is a deficiency of quality of protein in that diet, and repro may be impacted as well. So to finish up here, my take-home message today is that, hey, I think amino acid balance is here to stay. We very good ability because we have products available for methionine and lysine, but under an adequate amount of metabolizable protein. I can't stress that enough. If you go back to that 800 grams and add amino acids, maybe you're not going to see anything on the impacts, right? So it seems like increased milk yield, milk composition, oxidative stress, lower embryonic loss. I would say check maybe in that prepartum diet according to the energy that you're feeding. What are the grams of those metabolized amino acids that you're feeding? Not only the association with energy, and also that high protein or the average protein for the breed that the cows are showing are also associated with reproductive success of your farm. With that, I'd like to thank you. And here are my contacts, and I'll be glad to answer any questions if we have some time.
And we do have some time. Thank you,
Good.
Dr. Cardoso. Before we get started with the Q&A portion, we'd like to share a brief video. Then we'll be right back to answer the questions submitted during today's presentation.
Balchem has invested decades perfecting the art of nutrient encapsulation. And now, new AminoSure XL is setting a new standard, delivering 35% more metabolizable lysine than leading competitors. With AminoSure XL, you can provide your cows with a more consistent and higher quality source of metabolizable lysine, leading to improved and more dependable results. A recent meta-analysis found that feeding rumen-protected lysine led to an increase in milk fat percentage and 4.4 more pounds of energy-corrected milk. Feed new AminoSure XL to your cows and have confidence that you're using the most consistent, reliable, and cost-effective source of lysine on the market today. Learn more at balchem.com/XL.
Now, as a reminder, you can still submit questions through the Q&A tab at the top of your screen. Dr. Cardoso, we've had a lot of questions come in about the embryo quality and pregnancy loss. So kind of compiling several different questions. Why do you think there was a different response in multiparous versus primiparous cows in terms of embryo size and pregnancy loss?
Yeah, I think that's a very good question. Thank you for that. One of the things that I would think of is that, remember that primiparous cows, and I think I'm just going to put a plug here. I think we don't study her enough. We always focus on multipairs, and again, it's a question of number of animals, research resources that we have to allocate to, specific projects and to get answers. But I believe that, that first lactation cow, she's still holding off a lot of that milk yield, and therefore the requirement for that amino acid may be lower for that animal. When you have a multiparous, now you have a higher intake, you have a higher milk yield, and therefore a higher excretion or necessity of synthesis or protein synthesis for the mammary gland. So I think perhaps in the primiparous, there is not that increased usage of the methionine for the milk protein, where in the multiparous we have that that direction of the methionine for the protein. And remember that we kind of target our cows or design our cows genetically to really put everything to milk and not necessarily to get pregnant, right? So I think we are coming back a little bit now with genomics, and we are getting, I think, that story a little bit better, but I think that's why those results show there.
Excellent. Thank you for that. Next question comes from Sean. It says, Dr. Cardoso, great presentation, very thought-provoking. In the pre-calving diet, what gram of metabolizable protein and amino acids would you target? In terms of amino acids, is it just lysine and methionine, or do you target other amino acids as well?
That's a very good point. Thank you, Sean, for the question. So, like I said, the prepartum diet, it could be one group or pre-fresh close-up minus 28 days. I would recommend a minimum of that 1,200 grams. Doesn't mean that you can go to 1,300 grams and 1,400 grams. Some people, depending where they are in the world, they feel hard to get to that 1,200 grams, especially without having resources like blood meal, soy expellers. So we're going to have to use some of those products to deliver all the other amino acids that we are not maybe pinpointing yet. So I do look at that 34, 35 grams of methionine. I pair that with about 95, 100 grams of lysine, and I see what the diet is giving me, and then I just have to supplement or add the rumen-protected amino acids to it. Now, if you have blood meal, you're going to need less rumen-protected lysine, right? So it's going to depend a little bit of your resources. But also I've seen farms that are pushing 1,600, 1,700 grams of MP. And of course, everything is excess there. but then the nitrogen is stored in that cow. I think there is some compromise on the nitrogen efficiency and even energy balance of that cow that needs to be, just cautious, I would say. And I always look to the histidine story. And, based on INRA numbers, they kind of match our numbers. It's like whatever you have methionine, you should have histidine. But in the diets that I do here, I don't see any problems with it. But again, I have blood meal. Some places they don't have it. But It can be frustrating when you can't manipulate. Say, I know it's missing, but I have nothing to do. So then we ask Balchem, hey, can we have histidine? And then we ask Balchem, can we have arginine? And then we go.
Well, keep your ears open for that. One other question that's kind of along the same line is from Renan, and he says, is it possible to achieve good methionine levels by only providing corn sources and cow feed? And similarly, is it possible to achieve good lysine with only high RUP soybean meal sources?
I haven't been successful to achieve those levels that I'm talking about, only with the feed ingredients. I'm using a lot of those soy products that you mentioned there, blood meal, and still you need a little bit. It's going to depend on the amount, right? Mm-hmm. Sometimes we need a lot. Sometimes we just need a little bit to get that 35. And one thing I want to highlight is, like I showed you in that trial, we went from 26 methionine to 35 or 33 methionine. That sounds like a little bit like, oh, it's only 7. But that's a lot. On that metabolizable amino acid getting to the cow, that makes a big difference in how cows can, well, you saw it, maintain body weight or body tissues and provide milk components as well. So I think it makes a difference. So have been successful achieving these numbers just with the this products. Mm-hmm. I need those room protected amino acids.
Perfect. Thank you. Next question comes from Adan. Postpartum fertility and health-wise, do you believe anti-inflammatory substances can have a complementary role when feeding essential amino acids in the peripartum cows? Is there any scientific evidence to that?
I think there was some work here from, Kansas State at the time on feeding some anti-inflammatories at the beginning. However, there's a lot of, more recently, there was a lot of legal issues where some of those type of anti-inflammatories, especially more manipulated pharmaceutical, they were not being allowed to dairy cows or lactating cows. So there's a challenge there. I see that there's a lot of opportunities on maybe natural essential oils, other things that can act as it. And I think every time you can spare amino acids to be used for reduction of oxidative stress, and you can have the amino acid going for protein synthesis, that's a good idea, right? Mm-hmm. So I believe that's a good association. But again, us feeding NSAIDs or something, or adding to cows, that's not something that is not well received. But we could try to find some more natural organisms.
Sure. Next question comes from Ryan. Can you please reiterate what levels of methionine and lysine you'd recommend feeding to the transition cow? That's the first part. And then besides milk yield, what metrics should we be monitoring to verify the success of amino acid balancing?
Oh, that's a very, very good question. So let's come back to the recommend. So I would say like the dream, right, would be, or what we would like, that 35 grams of methionine on the prepartum and about 100 grams of methionine or of lysine. That would be the top. But remember, if you are at 20, maybe push to the 25, and then you get to the 30, right? And always think about your economics. You need to be formulating, optimizing for least cost, and all that needs to be done, right? So that's prepartum. Then postpartum, I would push the lysine to that 180 for that fresh period. And then I would associate with the energy to that methionine. And then I'm going to miss how much, what was the grams? Let me see here. That we were on that fresh period. So we were about 57. So I would say about 60 grams of that methionine at least on that very fresh period. And then you associate with energy. And I think we talk about 110 grams of methionine, 2 megacal of energy. And we were talking about 3 at least grams of lysine to the energy. And then the second part of the question was?
Let me go back to it.
One was
besides milk yield, what metrics should be monitoring to verify success of amino acid balancing?
Yeah, good point. I think that's just milk yield. It's not fair to the cow and to the nutrition because she's putting a lot of energy to create that protein and fat. We have hosting cows today here in the US that are producing almost like jerseys, and that takes a toll on the cow. So I think energy corrected milk, it's going to be a better would be a better indicator of success. And of course, that's what cows, everybody wants this in the transition period. Cows to give milk, so energy corrected milk, cows that do not get sick, and cows that get pregnant. So you need at least one benchmark or number for each one of those. Then depending on the farm, it may be no ketosis, no DA, no BHB at day 7 or 14. Repro, conception rate at first breeding needs to be over 40% for sure. Energy corrected milk, this is how much we want, you know?
Yeah. So it's consistent or it's subjective for each operation too.
Yeah, I think that needs to be that because each farmer will have a different goal, and you need to have that sensibility to perceive that and push little by little.
Daryl has kind of a question on the other end. How late into lactation, how many days in milk, should supplemental lysine and methionine be fed?
Okay, that's a good question. But again, let's go back to cows. They will need lysine, methionine, and all the other amino acids throughout the whole lactation, throughout their whole life, right? I think your question is like, I understand as when it starts becoming less needed for some processes. So the ingredients that I have in the diet will take care of it. I think that's the point. They need lysine, methionine the whole time. Mm-hmm. However, you find that they have a harder time consuming those amino acids during the fresh period up to maybe when they get pregnant, post-peak, when they move to the mid to late lactation. Perhaps at that point, they're producing low milk or lower milk that they don't need that. But again, you have to put that in the model, and the model will indicate to you how much is that missing. But again, some cows may need the whole time, depending if you have very poor sources of protein, you may need that the whole time.
Sure. Well, we have reached the top of the hour, and I know you said that you had another meeting to go to, but we've got one more question that I think is really a good one here. You used the terminology lysine pre-calving primed the cow for performance. If you were rationing a pre-calving cow, would you target higher lysine? And if so, how high would you go?
I would go to the 100 grams. I think, and then I'd say priming. Again, remember I mentioned, hey, MP, even in NASA in 2021, is not accounting for colostrogenesis that is happening in that pre-fresh period. So, and I'm combining maybe a little bit of information from the methionine trial, the heat stress, where we check the alveoli and amino acids do have an impact on cell regeneration, right? So what we believe when I say priming is that during that colostrogenesis, giving the cow all the amino acids that she needed, she was able to prime the mammary gland or have a larger amount or just a better quality type of alveoli. Again, that's why we didn't do a mammary gland biopsy to really tell you that, right? And then when she calved in those first 4 weeks, she was able to deliver that. So I think colostrogenesis, the same way as when we're drying a cow and she's reducing or going off lactation and needs to regenerate that tissue. I think those are very important situations that maybe we don't pay enough attention. So that 1,200 grams NP, getting the amino acids also includes something that NASEM is not considering, making sure the cow can have that colostrum, not just the colostrum, but also sustain early lactation. That's the point. And I'll go to 100 to see what happens there.
Yeah. We do have a lot of questions, and I'll put in a plug for our podcast series because we'll be recording a podcast here coming up soon to answer a few more of these questions. So
Cool.
Hopefully everybody can join us then. But thank you, Dr. Cardoso, and thank you everyone for attending today's webinar. If you do have additional questions, please submit them to anh.marketing@balchem.com. The Real Science Lecture Series continues with educational topics each month, including our next one on October 14th with Dr. Stephen Ashmead, who will present a forensic look at chelated minerals. Visit balchem.com/realscience for details on future webinars and to register for upcoming events. Balchem's podcast series continues to offer a deeper dive into our webinar topics. Log on to your favorite podcast platform and search for Real Science Exchange or visit balchem.com/podcast. If you want a cool Real Science Exchange t-shirt, just subscribe to the Real Science Exchange and send us a screenshot along with your address to anh.marketing@balchem.com and we'll get that off to you. On behalf of Balchem and Dr. Cardoso, thank you for joining us today.
Thank you.